Beneath every breath, oxygen leaves a small, sharp shadow behind. That shadow is hydrogen peroxide, clear as water, yet chemically restless. It forms wherever living cells harvest energy from oxygen. In leaves, roots, mussels, microbes, and milk, it appears quietly. A little peroxide can signal, too much begins to scorch. It nicks membranes, clouds proteins, and frays the chemistry of life. For early oxygen-breathing cells, peroxide was a locked and constant problem. The solution was already waiting inside nature's oldest laboratories. Bacteria, fungi, plants, and animals evolved a remarkable guardian, catalase. Wherever oxygen is busy, catalase is often nearby, standing watch. Its task is simple to say and astonishing to watch. It takes hydrogen peroxide and parts it into water and oxygen. No flame, no harsh metal bath, only a folded protein chamber. Inside that chamber, an iron heart meets the unstable molecule. Peroxide enters, splits, and leaves as safer, familiar substances. The sudden bubbles are oxygen returning to the air. A cut potato foams because its cells were full of catalase. Blood bubbles for the same ancient reason, red cells defending themselves. Long before the enzyme had a name, the fizz told a story. In 1818, Louis-Jacques Thénard isolated hydrogen peroxide in Paris. He saw it vanish when it touched blood and plant tissues. Chemistry had found a substance that living matter could quickly disarm. Yet the hidden agent remained nameless through much of the century. 19th-century benches filled with flasks, feathers, organs, and questions. Investigators watched oxygen rise from peroxide, but not from ordinary water. The reaction seemed alive, delicate, and easily damaged by heat. These clues pointed toward the young idea of enzymes. By 1900, Oscar Loew gathered the observations into one identity. He gave the peroxide-splitting enzyme its enduring name, catalase. The word carried the broader promise of catalysis itself. Catalase became a doorway into how living chemistry accelerates change. Scientists soon found it concentrated in liver, blood, and many microbes. In laboratories, liver pieces released storms of bubbles from peroxide. The ordinary organ became a teacher of invisible protection. Early researchers learned that catalase was not a vague life force. It could be separated, purified, and compared across organisms. The enzyme joined a larger revolution in biochemistry. Proteins were beginning to emerge as the working machinery of cells. In 1937, James Sumner and Alexander Dounce crystallized catalase from liver. The crystals showed repeating order, not mystical vitality. Catalase could be studied as matter, precise and beautiful. This helped strengthen the case that enzymes are proteins. Later work revealed the iron-bearing heme groups at catalase's core. Those dark molecular centers gave peroxide a safe place to change. One catalase particle holds several working chambers together. Each chamber passes peroxide through a narrow molecular doorway. The first peroxide gives up oxygen power to the iron center. The second peroxide receives that power, becoming oxygen and water. What might harm a cell is transformed in two quiet steps. The enzyme itself emerges ready to begin again. In living cells, catalase gathers in tiny compartments called peroxisomes. There, it patrols chemistry that naturally produces peroxide. Seeds use it as stored oils awaken into growing shoots. Leaves rely on it during bright days of overflowing sunlight. Microbes carry it as armor against oxygen and hostile surroundings. For humans, it works silently in liver, kidneys, skin, and blood. Its old laboratory drama now appears in many practical places. In food processing, catalase removes leftover peroxide after careful sanitation steps. In dairies and egg handling, it helps restore gentler conditions. In textiles, it clears peroxide after bleaching before colors are added. This avoids repeated hot rinsing and reduces the burden on water. In pulp and paperwork, it contained peroxide before later stages. In contact lens care, catalase helps neutralize peroxide into harmless byproducts. In wastewater, it helps industries soften oxidants before discharge. The same bubbling reaction becomes a signal in biosensors and test devices. Industrial catalase today often comes from carefully cultivated microbes. Fermentation can grow enzyme helpers without mining or harsh chemistry. The story that began with Parisian peroxide now belongs to greener industry. Catalase reminds us that nature's smallest tools can solve urgent problems. In its bubbles, chemistry points toward a cleaner, more patient future. Enzymes Bio exists because good science should be simple to source. We're enthusiasts who put the world's enzymes in one place for startups, labs, and B2B buyers. Kinder chemistry, here for generations to come. Find us at enzymes.bio.